Thanks, Joel. And thanks also for putting together this super interesting meeting and also providing an opportunity for me to meet old-time colleagues. I've spent 6 years at the Laboratory of Molecular Biology in Cambridge that were immensely inspirational and influential. And I guess the first thing that an Israeli learns when coming to Cambridge is that few drops of rain cannot paralyze an entire country. So my interest is really protein evolution. We've seen all these wonderful structures that perform these wonderful functions. And what my group and I are aiming to learn is how this complexity was generated by this evolutionary process of mutation and selection, small gradual changes that eventually led to this entire diversity. And basically, if I want to put in a nutshell the lessons that we and others have learned and that we've seen also in Madan's lecture, is that protein evolution is largely the recruitment of pre-existing structures of folds and active sites architectures to perform new functions. This is a process of tinkering, the foundation of which is based in this slide. And this is an hypothesis that I began to formulate when in Cambridge with Leo James, who was really the first group member or a member of a group of two, Leo and I. And the idea is that conformational diversity or dynamism of protein has, of course, functional implications that we've seen here, but also has fundamental evolutionary role. And that is to say that proteins coexist as an ensemble of structures, one or several of which are crucial for their native function, for the function that they've evolved to perform. However, there is this whole range of coincidental promiscuous conformations that do not relate to the native function of a protein. And it is this repertoire of conformations that may occasionally also encode promiscuous coincidental functions that evolution uses as a starting point if and when new function is needed. So while Leo was rising to the ranks, here with Her Majesty, I came back to the Weizmann Institute to study the details of this tinkering process. How, given a starting point, by gradual changes in sequence in the form of mutations, structure and function change to give you a new enzyme. And we have learned a great deal about this process, and I will not go into detail. I will just say that it turns out that actually teaching old dogs, meaning old proteins, new tricks, is actually a relatively easy process that nature and even us can reproduce in the lab quite readily. And this process has given rise to what we call today enzyme families and superfamilies. And Madan has shown clearly that even without changing actually structured regions which typically encode an enzyme, nature could generate this immense complexity and rewire existing proteins to generate like new organisms and new phenotypes. And this, as I said before, we know quite a great deal about how evolution kind of gave rise to orthologs and paralogs, or families and superfamilies. However, what we really don't know much about, or almost anything at all, is how the very first member of each what is now a gene superfamily emerged. Or in other words, how did the old dogs came about? How did the first dog emerge? And this is kind of has been I would say, the focus of my research in the last decade. And unfortunately, unlike the work of Madan and the work of Sarel that is to follow, that is of like huge utility, potential utility, I'm afraid that this work is entirely, utterly useless. Its only purpose is really to satisfy our curiosity, to understand really how these proteins came about, and in coming about enabled us to basically be here, think and talk. We also don't use machine learning, only human learning, which is painfully slow. So how did the first protein emerge, or the first proteins emerge? Basically, the first hypothesis in this area was formulated by this amazing scientist, of whom few people unfortunately know, Margaret Dayhoff, who is the mother of bioinformatics. If you're using the single letter code of amino acids, which you are, the alphabet, if you're performing an alignment or a tree, you're using tools that were envisaged and developed by Margaret Dayhoff. And in this seminal paper in Science, Margaret Dayhoff suggested that proteins, the first proteins, emerge by duplication and fusion of short peptide segments, whereby this duplication and fusion would provide the volume and complexity that a protein needs in order to perform biochemical function. And basically, this hypothesis was found support in the fact that many protein folds show internal symmetry. So they look like the products of duplication, but there was no experimental support for this hypothesis. And the first one came from the work of Robert Smoke in my lab, who followed the evolution of this highly symmetrical, beautiful fold called beta propeller. So this is a lectin, a sugar-binding protein, that Rob could show emerged by co-option, by stealing a fragment of about 50 amino acids from a protein that has a completely different structure. And by duplicating this segment 5 times and fusing it, a new protein emerged. However, a critical intermediate that was revealed by this work is that the first step in emergence is actually oligomerization of this segment to a pentamer to give a protein which is essentially identical to wild type. So we propose that actually oligomerization, and as you will see later, even simpler forms of self-assembly are the key to the emergence of complex proteins from short segments. And we overall propose this mechanism by which a polypeptide fragment emerges, either from co-option from an existing protein or even from a non-coding region, or by maybe spontaneous hydrolysis at the early stages of life. This fragment oligomerizes initially or self-assembles to give you sufficient structural volume and complexity, and later a series of events of duplication and fusion give rise to mature globular proteins. The snag is that while this protein tachylectin is of interest and beta propellers can do nice things, they are very recent inventions in evolution. These proteins have largely emerged in the last 500 million of years, which means essentially a second ago on evolutionary time scale. What we are truly interested and know almost nothing about is how did the very first proteins emerge. And this is an event that had occurred on this planet about just 500 million years after it emerged. And by the time the last universal common ancestor was here about 4 billion years ago, it already had a full set of proteins that are very similar to the proteins that E. coli and us carry today. So really, if you are a protein evolutionist, nothing interesting happened after LUCA. All the excitement was there when we were, sleeping or still in the planet. So how did the very first enzymes evolve? I'll try to give you a glimpse on that process. What we, others, and we assume that this emerged, and I touched on this, on a world that was kind of on transition between RNA and proteins, and specifically something that people have dubbed the RNA peptide world, where RNA cooperated with amino acids and short peptides that could basically complement the function of RNA and provide it with stability and other enzymatic activities that RNA could not provide. And the assumption is that from these fragments, or peptides, sorry, polypeptides that emerged in the RNA world, modern proteins have evolved eventually by this process of duplication and fusion. And basically our work aims to try to establish this scheme and provide some details about this process. So in order to basically understand a bit more, we would need to walk back from today's proteins and to try to infer on what could have been the first peptides. And to do this, we first need to understand what were the first enzymes doing, what were these first enzymes. And previous analysis and a systematic analysis done by Liam Longo, a postdoc in my lab that has just been published 2 days ago, suggests that the first enzymes bound and utilized phospholigands, various ligands from RNA to ATP and other like nucleosides that contain phosphate groups. And there are specifically 2 superfamilies or folds that come up time and again in these analyses as the earliest enzyme folds. And these are the Rossmann and the P-loop domains. So here are 2 major contributors to understanding of protein world. Michael Rossmann, who sadly died last year, who identified the first enzyme that belongs to the Rossmann superfamily, lactate dehydrogenase, and John Walker, who was at the LMB, who worked on ATP synthetase, who is just one representative of a huge superfamily now called the P-loop NTPases. One cannot overstate the importance of these 2 superfamilies, to which most of the enzymes that mediate core central metabolism belong. In fact, if you go to like simple genomes of nanoarchaeota or simple bacteria, up to 40% of the proteins with known structures belong to these 2 superfamilies. So it's nearly half of the genome is either Rossmann or P-loop NTPase. And another unique characteristic of both these folds and the other folds that are like that this analysis has highlighted as the most ancient is that they bind phospholigands in a unique manner at the tip, at the end terminus of the alpha helix. Without exception, all the ancient folds have the phosphate stuck, if you like, as a plug at the end of at the end terminal of a helix. And why is that? Perhaps this is the easiest way by which evolution finds phosphate binders. Turns out that not. Liam's analysis has looked basically at all the incidents throughout 4 billion years of evolution where phosphate binding emerged. And turns out that there are about 300 such cases. 300 times independently, evolution has come up with a protein, new protein that binds phosphate. However, a small minority, as you see here, use the end terminal of a helix to bind phosphate. So why did the first proteins chose, or had maybe not any other choice, but to bind at the end helix? The answer is that if you look at simplifying constraints, meaning if you ask the question, can a phosphate binding site emerge within a short segment of sequence, not comprising position 20, 200, and 350, but rather a short segment of primary sequence that is also comprised of what we call abiotic simple amino acids, then the N-helix is the only solution that meets these constraints. It's the only way by which a phosphate binding site can emerge within a segment of several amino acids that are, say, glycine, serine, and threonine. And this analysis has further pointed out these two segments that are highly simple. They comprise basically a beta strand, a short glycine-rich loop, and an alpha helix coming from the Rossmann fold or from the P-loop antipiases, where this is called the P-loop or the Walker A motif. And the Rossmann fold has a very similar phosphate binding loop that is actually glycine-rich, and we believe, by the way, that the two are highly related, but I will not dwell on this. It's a kind of controversial and old question in protein evolution. But both these folds share the simple structural motif that has a single functional element, which is a phosphate binding at the end terminal of the helix. And basically, about 12 years ago, my lab began to try and find experimental support that these elements can do something, anything that could have been useful at the emergence of life. And the first step was bioinformatics, was actually data mining, so it's unavoidable. We have millions of sequences of P-loops that belong to very different structures and have very different different functions. However, they all share this motif. And Agnes Petrovsky has basically elaborated this mega tree, if you like, and they've extracted a logo or inferred a logo of the ancestral P-loop sequence, where you see this beta strand here, the Walker A motif, and the helix that follows. And remarkably, although you cannot align these proteins anymore, this segment does align and does give you reliable inference. And the second thing was to try to then experimentally test this. And after a daunting period and 2 or 3 people abandoning the project, eventually we had to surrender to computational design. There is nothing more humiliating to a protein evolutionist than to use computational design, but occasionally we do it. And some of my best friends are computational designers. So actually, upon visiting David Baker, David said, look, we have these very simplified ideal folds that actually mimic or are simple mimics of the Rossmann and the P-loop. These are what are called alpha beta-alpha sandwiches, where this is a parallel beta sheet, exactly like the Rossmann and the P-loop NTPAs. And what we have done was extremely naive. We took this ancestral motif, and with a bit of Rosetta tinkering, 2 beta loop alpha elements were substituted for this ancestral-like motif. So we basically had a protein of about 100 amino acids that contain 2 P-loops and relates to this sequence. And lo and behold, something close to a miracle, this protein was found to bind ATP and, more surprisingly, to avidly bind RNA and single-stranded DNA. And he even have very weak ATPase activity. So this was basically the first breakthrough that showed that even in a relatively simple context, the P-loop can bind phospholigands and can have a biochemical function. And this work that was initially initiated by Sisi or Maria Luisa is now being continued by Pratik Vyas, a postdoc in the lab, whose results I will show in the rest of the lecture. And what Pratik has asked is, can we make this protein, which contains 4 beta strands, 4 helices, and 2 P loops, even smaller and simpler? So what Pratik has done is to do a series of manipulations of cutting different fragments, the N half, the mid half, or the C half of this protein, and performing some circular permutations to find what is the smallest and simplest fragment that is still functional. And basically this has converged to this protein of 45 amino acids, which is the ancestral beta strand P-loop helix, flanked by, at the end terminus, by 1 helix. And this protein turns out to be functional. And the second thing that Pratik was looking for is a function that would be more meaningful than just binding ATP or DNA. Something that the first proteins could have been doing or were doing in relation to the RNA world or to the RNA peptide world. And we know that P-loop NTPases do wonderful things. For one thing is to synthesize ATP. This is how this fold was discovered. But they also do a lot of curious and interesting things with DNA and RNA. Actually, most of the family members are motor proteins, if you like, helicases, translocases, recombinases that massage DNA and RNA and allow them to function. The snag is that these are dauntingly complex proteins. So if you look at the simplest helicases, there are these hexameric structures that go through 20 different conformations in order to act as helicases. Or take, for example, REC-A, the recombinase, the universal recombinase. This is extremely complex machinery that can separate double-stranded, unwind double-stranded DNA, do strand exchange, mediate the Holiday junction. It can be present as monomer, dimer, hexamer, or a filament. So can we basically reproduce any of the functions of a molecule of the complexity of REC-A in a simple peptide? If I would give this as a question to undergraduates and they would say yes, I would fail them in the exam, right? Because how can you crop basically a segment of 40 amino acids from this complex machinery? This is the monomer. This is even the REC-A monomer, where it also forms an active site that is excluded from solvent, and think that this could do something which is even remotely reminiscent of REC-A. But turns out that it does. So the first thing that Pratik has tested actually is to see if we can unwind double-stranded DNA. This was based on the observation by Sisi that these proteins preferentially bind single-stranded DNA and RNA over double-stranded. So we use this beacon assay by which you have a fluorophore quencher here that are, when separated, the molecule is fluorescent. And if you have something that will unwind this double-stranded DNA, it would collapse into this hairpin and fluorescence will be lost. And basically, lo and behold, as you take this double-stranded DNA and you add this fragment, there is an immediate loss of fluorescence, indicating that it is collapsing to this hairpin. And turns out that unwinding is driven by highly avid and highly cooperative binding to single-stranded DNA. So basically, these fragments bind with micromolar, submicromolar affinity. And as you will see, I will return to this, the binding is highly cooperative with the Hill coefficient by this fit at least of 4. It turns out that basically all you need is this ancestral fragment that you have seen of a beta strand, Walker A or P-loop, where the phosphate binds here and this helix, and just some part of that, like N-terminal, if you'll be like solubilizing tag. We don't think that this part has a structure or a particular function, because actually you can truncate most of it and retain the function. And mutations in the P-loop abolish binding. So basically the function of this fragment is mediated by the Walker or the phosphate binding loop. But so far what I've shown you here, this strand separation, is something that any single-stranded DNA binding protein could do, right? It's just shifting the equilibrium from double-stranded to single-stranded DNA. But can these proteins do something else? We then took advantage of the fact that they seem to bind ATP, or they bind ATP. And indeed, so what you see here is double-stranded DNA. The protein is added, and it collapses to this hairpin. But then if you add ATP, you reverse the process. Basically, ATP will displace the DNA, that will in turn relax back to the double-stranded state. And further, it's not only ATP. This is one of the other curious findings. Because GTP actually does it better for reasons we don't understand. But foremost, these proteins love inorganic polyphosphate esters, such as hexametaphosphate or polyphosphate. And polyphosphate was actually suggested by Arthur Kornberg, Roger is not here, who identified the enzymes that make polyphosphate and suggested that this was the precursor of ATP, that this was the first source of biological energy was inorganic polyphosphate. And these proteins simply love polyphosphate. And if you look at this phospholigand-driven release and realigning of the double-stranded DNA, you can see that hexametaphosphate does this with a micromolar affinity, and ATP and GTP do the same, yet with millimolar affinity. And again, to remind you, this is a peptide of 40 amino acids with a single functional element. But REC-A does not only like separate or unwind double-stranded DNA, but it can also do strand exchange. So can these fragments do something similar to that? Because so far, as I've said, I've shown you double-stranded DNA unwinding, which is driven basically thermodynamically by shifting the equilibrium owing to the higher affinity to single-stranded DNA. But you can also do the reverse, meaning you can take this hairpin, where the fluorophore quencher are here, so you have no fluorescence, add a competing single-stranded DNA that has no fluorophore quencher, and this process is already thermodynamically favored, right? Because this is double-stranded is more stable than the initial state. And indeed, if you do this in the absent, just in buffer, you will see increase in fluorescence that corresponds to the, basically to this process. But this exchange is relatively slow. If you add these fragments, you will see instantaneous exchange, indicating that they also accelerate, that these ancestral P-loop fragments accelerate strand exchange, meaning they don't only do a thermodynamic shift, but they can act to accelerate a thermodynamically favored process. So you might ask, and we are still puzzled, by how a polypeptide with a single functional element can do all these things, which we don't expect peptides to do. And the answer really goes back to this scheme. It's owing to its conformational plasticity. So the first thing to say is that these proteins are ordered. They are not disordered. If you take them to CD, they show clear signal of beta-alpha structure. And indeed, the group of Gabriele Varani in University of Washington was able to solve the structure of just one of these proteins that was monomeric on a good day. And you could see that they could identify at least 2 coexisting conformations, where you can see that this is the P loop here, is like this is the major conformation and this is the minor one, but you can also see differences in the helices of the 2 coexisting conformers. But this is just the tip of the iceberg, because it's not just the secondary and tertiary structure of these proteins that is so highly plastic. It's foremost, it's their quaternary structure. As we've seen, say, in other talks or in China's talk, it's like most proteins that we see in the textbook have a predefined oligomeric structures, but some of them have this ability to fluctuate between different states, going all the way from a monomer to a fibril or to a polymer, as you will see here in a moment, and the same holds for here. So the key is also oligomerization or even simpler forms of self-assembly that enable this simple polypeptide to perform relatively complex functions. And indeed, cross-linking shows that this is not a monomer. This protein is likely to fluctuate between something of a dimer up to a hexamer when it's free in solution without a ligand. And when you add a ligand, this is the DLS, dynamic light scattering, there is an abrupt and dramatic change in size. So this is the protein in the absence of a ligand. You can already see that its size is anything but the size you would expect from a 45 amino acid peptide, including the histag. Upon addition of hexametaphosphate, there is a transition to a larger structure, the details of which we have no information at the moment, but it's clearly polymerizing into some kind of a fiber. And actually, if you titrate the phospholigands as Pratik has done, you see basically the same picture that you have seen with the release of the single-stranded DNA, meaning hexametaphosphate induces this transition with very high affinity and to a higher, larger like species, whereas GTP, for example, induces a change, but to a smaller species and with lower affinity, suggesting that the binding events that we see in the strand exchange assays and the transition in the DLS correspond to the very same event, meaning the binding of phospholigands to these fragments. And sodium phosphate that cannot induce strand exchange also does not induce like this transition in structure, indicating again that this is a specific binding of these ligands to the P loop that can induce these transitions. So basically in this small fragment, we see kind of the hallmarks, if you like, or the fingerprints of what its cousins that have evolved much later can do. Molecules like REC-A. We see this in the ability to unwind double-stranded DNA, to mediate strand exchange, to bind both RNA, DNA, as well as small ligands such as ATP or GTP, but also to basically transit, if you like, between all these states, which are monomers, dimers, hexamers, and possibly filaments, as the DLS suggests. And I must say that this research had periods that were extremely daunting, where we were just looking at the dark and how can you basically put this ancestral fragment in a context that would do something, anything. And there's only in the last, I would say, 2-3 years has given the fruit that I've just shown you. But this has been extremely rewarding. And then these wonderful words of Darwin echo in my ears whenever I see the results coming from these experiments, of saying that there is a grandeur in this view of life, from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. So what I was trying to do today is to give you a glimpse of these so simple a beginning. And to summarize our findings, I would say that the simple beginnings involve polypeptides that bound phospholigands, a range of different phospholigands, where the tip of a helix comprises the hotspot where phosphate binding has emerged and still is retained to this day. These fragments were not only small or relatively small, but were also simple. I didn't have time to talk about it, but essentially these proteins have not a single aromatic amino acid or histidine. They are all made of what's called abiotic amino acids. The only non-abiotic amino acid is actually the lysine of the Walker A motif. The key to function in small fragments is like the functional versatility on the one hand. This remarkable, the modern P-loop binds only ATP or GTP. Modern P-loop proteins do not bind, I mean, they bind DNA and RNA, but by other motifs, not by the P-loop. The P-loop only binds ATP or GTP. But these primordial fragments have this versatility that the modern P-loop has lost in being able to bind not just NTP or GTP, but also polyphosphate, RNA, and single-stranded DNA. And in this conformational plasticity, including the plasticity of quaternary structure and this ability to form oligomers, as well as like more rudimentary forms of self-assembly. And finally, I did not have time to speak about another work that is now in bioarchives, where another glimpse of the emergence from a simple beginning of a very common fold that mediates DNA and RNA binding called the helix-hairpin helix or HHH motif, which is seen in now zillion of proteins that work on DNA and RNA, where actually we also could provide some hints of what was the first basic amino acid, suggesting it was ornithine, actually, that was chemically converted to arginine. And also phase transition has been discussed here, and this is how these droplets look like, suggesting that the earliest precursors of this fold were actually interacting with RNA through phase transition and have later adopted this helical structure and specific double-stranded DNA binding. Okay, so finally I want to thank or acknowledge the group of dedicated people that, unlike myself, do experiments. I haven't done anything of the experiments shown here, and don't just go from one conference to a mountain to a conference to a mountain. So the P-loop work has begun with Maria Luisa Romero, a postdoc in the lab, and is now being continued by Pratik. And Liam Longo has been behind much of the work we've done on the early emergence. I've shown a bit of his bioinformatic work, but the ornithine work is basically like spearheaded by Liam. We are now fighting to get the paper through. And Dragana Despotovic and Jagoda Jablonska are also part of the pre-LUCA protein evolution team. And thank you very much for your attention.
So, Danny, thank you very much. I think this was incredibly elegant talk. Although he began very modestly saying it's useless, it's not clear to me it's going to be so, where are you? That it's so useless. In the sense that I think, Donnie, some new materials could, among other things, could come out. But let's open the talk for questions. Shut up. Yes, the microphone.
Really interesting. So your last part, just very simple peptide and mimic RECA, and show that really highly cooperative binding to single-strand DNA. That really indicates specific protein-protein interactions. So from your sequence, can you predict where the interaction might be? Is that by chance or by design?
By design, I wished. I mean, these proteins were initially designed as monomers. And David was disappointed to see them doing all these tricks. But I was, of course, ecstatic. I think the simple answer is we have no clue. So actually in the audience are two people, Dana and Ori, who have been trying to convince who are structural biologists to try to gain some insight. I think we don't, any guesses we will make will end up humiliated at the end.
Have you looked at the conformation of your protein in the oligomeric state? Is it still the same canonical alpha, beta, alpha? And the reason I ask is because Ulsher Jacob has this very nice work showing that polyphosphate drives amyloid formation for a whole variety of different proteins involving inducing conformational changes.
So far, embarrassingly, every time we take any of these samples to the CD, they show the same spectrum of alpha, beta, for whatever it means, because CD is CD, it's low resolution, but the helical nature seem to be preserved. So I don't think they fall into amyloid. Incidentally, our very first experiments were just taking this peptide. And this peptide was forming amyloids that were tantalizingly dissolved when you added ATP. And, I have days where I'm kind of tempted to go back to it, and then days where I say, just hammer on this. It's not clear to me whether we've seen, there was Tony Hyman showed that ATP is a hydrotop that actually can dissolve amyloids, or we are really seeing like something specific. Clearly amyloids provide a solution to a very simple form of self-assembly, as does phase separation or coacervate formation. And I think these are forms that should be considered as the earliest kind of forms of assembly of proteins to give you something functional.
Sounds like a good project for an NMR spectroscopist to me.
I wish the reviewers of our grants would think the same, but...
Question was answered. Other questions? Please.
Is this sequence is similar to GTP binding domains in proteins?
Yeah, this is what it is a proof of concept because no one, even someone as old as I am, was there at the point of emergence. But they are like clearly linked to modern protein, because if I give you this sequence and you will blast it, then REC-A, helicases, ATP synthetase will come up. It's clearly like and unambiguously linked to the modern proteins. Thanks. This is a good point. So it's in that respect, it's a bit more than a proof of concept.
Do these fragments hydrolyze ATP or GTP?
So the first protein that I showed that has these two P loops has consistently shown ATP hydrolysis. We were very careful because this protein is produced in E. coli. And to rule out contamination completely was not so feasible. But we've done any possible control and multiple purifications, and this ATPase activity persists. However, it is very weak, and the next step is probably, in order, we think that really that the p-loop has initially emerged as a binding element, as it is in the Rossmann fold, because in the Rossmann fold, phosphate binding is merely, it has no catalytic role. Catalysis occurs elsewhere. What you need to turn this into a catalytic, into an ATPase motif, is something called Walker B, which, if distilled to the simplest form, is actually an aspartate at the tip of an adjacent beta strand that would then ligate magnesium, and which is essential for the ATPase activity. And indeed, this is the next step in our drawing board, is to introduce this aspartate and try to convert these from binders to catalysts.
I wonder what happens if you try to reconstitute it into lipids. What happens? It goes into protoliposome?
We haven't tried that. Some of these, I mean, this last construct that I've shown, which is a short helix beta alpha, is actually relatively well behaving. Others require all sorts of tricks to solubilize them. We haven't tried lipids. It's I guess, a good idea. And maybe, I mean, I think Pratik has coined this term that what we can expect with these proteins is the unexpected. So, but it is worth trying. Because
the ATPase is in the membrane.
Yeah, and also, as I said, I mean, they are not monomeric. They need to stick to each other, or maybe to lipids. So that's actually a good idea.
Great. So again, we want to thank Danny for this great talk.